So in this practical class, you're going to harvest your experiment and collect all the data that you need for your experimental report. So all 32 plants are going to be returned to you, again in these two black racks that you can see here. And the first thing to do is reweigh all of the herbivores that you applied last week. So using the plants with red labels, carefully remove the cage like Kamina's doing here, and find your insect on the plant, which is situated on this leaf here. So carefully remove the insect using a paintbrush, and once Amina's done this, it could take a few seconds. Then take it to a weighing station, so this is more sensitive of the two balancers, and place it inside the weighing boat like this. And remember to make sure that you've gone to 0 before you take the weight of the insect. So in this case, it's around 0.072 grams. Once you've taken the mass of the herbivores, you can actually remeasure the plants then. So like you did in the first week, you can measure plant height with the ruler like this, and also count the number of leaves.
1, 2, 3, 4, 5, 6.
Remember to record all this data in your Excel worksheets. Now, stage 8 in your protocol says that you need to test for fungal endophytes. And a lot of these grasses contain these endophytes. Don't worry about that, but what we need to do is just make sure that the plants either do or do not have these fungal endophytes. And the way that we do this is to cut the plant at the stem, like Kamina's doing here. And you'll each receive a blotting paper with 32 numbers on like this. Now, using the corresponding plant number, so that's 27, what you must do is basically blot the leaf in that particular square like this until you can see that there's kind of a little bit of a gooey mess in that particular grid square. And do this twice. So once you've done the blotting test for the fungal endophytes, you need to measure the mass of the shoots, basically, which is a bit that you've just cut off. So go to a balance, make sure you re-zero it, and then take the mass of those leaves. So in this case, it's 0.29 grams. Once you've done that, find the corresponding label that corresponds to your plant, basically, and put this on a brown paper bag and put your leaf material inside that bag. Okay, so now we need to separate the roots from the soil in the rest of the pot. So what to do is basically lift the pot out of the rack and try and tip out the soil and root material as best you can, basically. So this is very sandy soil, so it should be fairly easy to separate the root system from the soil. Here we go. And once you've got the root system, like Kamina's done here, you've also got a bucket of water that you can use to wash away all the remaining soil. But try and remove as much as possible before going to the bucket. Once you've got your roots nice and clean, apply some paper towels to the bench and dab dry, basically. Now what you need to do at this stage is, once you've got them dry, is weigh the roots in just the same way as you did the leaf material. Once you've got your roots nice and clean and you've dabbed them dry as best you can, take them to the balance like you did with your leaf material and place it inside the weighing boat to record root mass. In this case, this is 0.36 grams. And in just the same way as you did with the leaves, the shoots, get yourself the corresponding label, apply that to a brown paper bag, and put the root material within the bag, and you're done. Remember, whilst you're going along and collecting all these measurements, to put them in the Excel worksheet that corresponds to your group.